Three more minutes. Let me share the PowerPoint so we can discuss cancer genetics. Right? Can you see the PowerPoint, everyone? Can you see the PowerPoint, please? Great. Great. And can you hear me all right? You can hear me all? Great. Everything looks good so far. We are on time. There's no waiting. I don't want you to wait. Let's be on time and finish it on time. I will do this for an hour because I don't want to rush over it and then we should be done, right? Because cancer genetics has, remember, 9 questions, right? And it's complex, believe me, not easy. So let's not rush over it, but yes, you can stay on after an hour if you have questions and you want to clarify. Otherwise, we should be good. Okay?
Dr. Ba, is this PowerPoint on Canvas?
Yes, this PowerPoint was shared with your class reps to share with you. I shared a PowerPoint with questions, practice question, and a PowerPoint. I think if Shireen is there, she can explain, right, Shireen?
So sorry, I'm sending it out right now. Give me like 2 seconds.
Oh, please. Please send it because I'm discussing the same PowerPoint. So it would be nice. Great. Yeah, I sent it to her. Your genetics, I will tell you later. Let's finish this. We have not even started the how many questions for exam. I will do exam review and tell you exactly later on. This is not exam review, okay? Got it? Yes, everyone? Yes, I will definitely tell you everything. I tell you the splits, right? I tell you the split of the exam, but not right now. Yes, we're just doing cancer. We are redoing actually cancer genetics right now so that you people have no confusion. There's a lot of confusion and some questions have not been addressed, right? So we are redoing the session actually. So this is not an exam review. But yes, when there is exam review, I'll give you all the split. What are questions? You know, there are only 2 sessions, as you know, right? It's a good grade grabbing exam. Please don't lose it. I think it's 10. So I would now, last minute people joining goes there. Let me make her a co-host. Okay. Thank you for doing that for me, Mr. Tahir. Because let's start now, please. Okay, first of all, I want this participation. This is being taken care of. Thank you. Thank you for doing that for me. Okay, now see, please, first of all, there is a clear communication of how the supplemental sessions are run every week. First hour is me, your class reps, and Diego, who's always as a CC in class rep because they were working together. That your first hour is by me and the second hour is by Dr. Chalagundula. Please, there is no confusion, there is no miscommunication in that. So moving forward, you know that. But to avoid this, I've changed things. There will be only 1 hour supplemental session now by me. If you have any Zoom, you wish to do any Zoom with Dr. Chalagundula, please email him. Tell him find out a time and Zoom individually. So we don't have this confusion anymore lurking because the course is almost over. I don't want any glitches. Okay. Is that clear, everyone? There will be only 1 hour supplemental now by me. If you need any Zoom, please email him personally and you can deal there. There should be no glitches. I don't want my course to have glitches. I'm very organized, clear-cut instructions. There's no confusion, right? Canvas is there. If there's anything, that will be changed. Now let's look at cancer genetics. Please, I want to review some questions because they were not answered and if there is any confusion. First of all, can you see my I have your chats open. If there is anything that I can quickly answer, I will answer. But I will actually concentrate my time in doing this session because remember, I'm redoing it, right? Is everyone having the slides that I shared, please? Everyone is good? Great. So let's see what we have to do. First, please stick to your learning objectives because they are tied to your questions. Answering learning objective will help. What I'm going to do is I'm not going to redo this session in eliminating every answer choice. I will redo this session telling you why this is the right answer. First of all, please, please, and please, when I'm telling you, note it down, go back and look at the video or the lecture slide. First of all, cancer is monoclonal in nature. What do you mean by monoclonal? It's a single cell that has the defect. What is the defect? There are two theories. One is the nuclear theory. There is gain of function of, tell me, proto-oncogene or tumor suppressor gene. First, you got to be clear with that. No flip-flop. So gain of function is for proto-oncogene or tumor suppressor gene. Proto-oncogene. Don't flip-flop that. And loss of function mutation of? And loss of function mutation is of? Now, somebody wrote oncogene. Now, let me clarify that too. Proto-oncogenes are normally involved in growth and proliferation. You need them. But proto-oncogene can be converted into oncogene that is cancerous. So when I ask you gain of function of mutation, you would say proto-oncogene. Does that make sense? Somebody said oncogene also. I'm correcting. Proto-oncogene will change into oncogene if it is. No worries, I'm just clarifying. So gain of function mutation, proto-oncogene. Loss of function mutation, tumor suppressor gene. Any confusion in that? Right? Don't flip-flop that, please. Secondly, please know that cancer is not one-step process. That gain of function mutation, loss of proto-oncogene, loss of function of tumor suppressor gene, and you are done. No, this is a multi-step process. Yes, it is a single cell, monoclonal in nature that has the defect. But remember, there's a mitochondrial theory also. What happens to mitochondria in cancer? Are they normal or defective? Are they normal or defective? They are defective and there is lactate accumulation. Lactate accumulation is favorable to acquire more and more mutation. So yes, first there is a monoclonal cell has a defect, either gain of function mutation of proto-oncogene or loss of function mutation of tumor suppressor gene. But so what is cancer? It's a tumor, it's a mass. How does a mass form? Yes, there is rapid proliferation of cells, but in doing that, they keep accumulating more and more mutations. But one thing, so it's not a one-step process, it's a multi-step process. But there is a single mutation, but then they keep acquiring more mutation. Yes, from exam point of view, when you do disease, you only need to know that single mutation, not the other mutations they have acquired. Why? Or collected? Because that single mutation is the cause of that cancer. That is very clear. For example, in familial adenomatous polyposis, what is the gene involved? There's only one gene. Can anyone answer me? Though it's a multi-step process, what is the gene involved in familial adenomatous polyposis, FAP? Just give me the gene. I'm not asking you to know the function. Okay, let me ask you, is it APC or is it MLH, MSH? Is it APC or MLH, MSH? FAP. APC. So the gene is still the same, but they will acquire more mutation as they proliferate. Now, last thing students don't understand. So yes, monoclonal single gene, acquire more mutations. Students don't understand that, which you don't have to memorize though, because there are so many mutations they acquire. But last thing that students don't understand is now this mutated cell should undergo what? Normally should undergo what? There are checkpoints in the cell cycle and mutated cell should undergo what? Apoptosis. So but what happens in cancer cell? They evade apoptosis. They evade apoptosis. So cancer is not just about proliferating a mutated cell, but also surviving it. So increased survival and increased proliferation leads to cancer. So keep both in mind. Secondly, so should you know apoptotic an anti-apoptotic gene? Absolutely. I will bring that later. So now let's do that. So have you understood cancer concept? Monoclonal? Yes, single defect. Whether gain of function, proto-oncogene, loss of function, tumor suppression gene. But don't forget, not one-step process, multi-step. When they are proliferating, they will accumulate more and more mutation. That's also not enough. They have to survive this mutated cell. They disregard checkpoint and they evade what? What do they evade? Programmed cell death, which is known as apoptosis. Are you clear with the process? So let's do this question. So I will tell you why this is the right answer. Again, I told you there are checkpoints. Should you know now who protects your cell cycle checkpoint? Yes, there are caretakers and there are gatekeepers. I will bring, yes, P53 is one of the answer, but I don't want to bring it here. I want to go stepwise with cancer. So what are these checkpoints? Checkpoints are controlled by genes. We'll talk about them. So should you know two genes that control checkpoint? Absolutely. Please write them. They are gatekeepers and caretakers. What do you mean by gatekeeper? Naturally, gatekeeper will not allow. What does the gatekeeper do? The gatekeeper will not allow the mutated cell to go into the cell cycle. What will the caretaker do? By chance, the mutated cell went into the cell cycle, but it got to be repaired. So what are caretaker doing? Repairing the mutation. So both are taking care of the cell. That mutated cell does not enter the cell cycle. So should you know gatekeeper, caretaker? Yes. So is this true that there are checkpoints, especially G1S phase? Please know that G1S phase is responsible for cell proliferation and progression. And what are those checkpoints? What did I tell you? Tell me the two checkpoints. What are those genes? They are either caretaker or there are gatekeepers. We'll talk about them. Now let's do one wrong choice because I want to concentrate on every question. So that's why I can't do every elimination. Now look at this. They are saying mitochondria is functional. That's not true. There are two theories in cancer, nuclear and mitochondrial. Although I have explained in my video that both theory exist, but I would recommend they have to combine these two theories because both go wrong. So are mitochondria functional in cancer cells? Yes or no? Are mitochondria functional in cancer cells? No. So that's a wrong answer, which is wrong, which is choice C. So I'm just explaining you more the right answer, please, because you have confusion in that, right? And explain you one wrong answer. Is this helping? Does this help everyone? Great. Now, monoclonal in nature, absolutely. Acquire mutation, genetic instability, just read them. But what are the three main points you should know for cancer? Is there altered metabolism? Yes. What will we discuss here? Warburg effect. You should know Warburg effect and PAT scan. Absolutely. Please note it down. We'll talk about what is the altered metabolism. There is lactate accumulation in cancer cells. Is it? Yes or no? Is there lactate accumulation in cancer cells? That is known as Warburg effect. Secondly, is there altered gene expression? Yes. Gain of function is for which gene? Gain of function is for which gene? Gain of function is for which gene? Proto-oncogene. And loss of function is for which gene? Mutation. Tumor suppressor gene. Is there altered motility? Absolutely, because cancer cells metastasize. Why is metastasis? Metastasis is the only key difference between cancer is there, but it can be benign and malignant. When do you call cancer malignant? When it's invasive. Please know that. So yes, there is an altered motility and cancer can become invasive. When cancer becomes invasive, is it benign or malignant? Is it benign or malignant? Is it malignant? Now, there is a term I have used in my video, which is carcinoma in situ. Now, please understand carcinoma in situ is also invasive, but cells have not invaded yet. So carcinoma in situ does not mean it's benign. It's still invasive, but cells have not invaded yet. So that can also be a condition, right? What do they invade? They invade the basal lamina. and then they go into metastasis. Do you need to understand that when I say cells are monoclonal in nature, what do you mean by that? For example, most common cancer is carcinoma. Do you know where they derive from? Connective tissue cells or epithelial cells? When you say carcinoma, they are the most common tumors or cancer, epithelial cells. That's why they say it's monoclonal. It's that epithelial cell that has the defect. Does that make sense? And which is the most common cancer? Carcinoma in epithelial cell. So have you understood the three key damage by cancer? Metabolism is altered, lactate accumulation. Gene expression is altered, gain of function, proton gene. Loss of function, tumor suppressor gene. And lastly, there could be motility. Cancers can be invasive. When cancers are invasive, they are not benign but malignant. Right? You can note down your questions if you have. Now let's understand what is Warburg effect. Now please tell me one thing, that when do you form lactate? There were two conditions when you form lactate normally in your body. When do you form lactate? Two conditions when you form lactate in your body. Tell me. That is an anaerobic. Oh God, I got a fly in my room. Sorry, but that means another student in my room. Yes, lactate accumulation is because of anaerobic glycolysis. But what is the cause of anaerobic glycolysis? Two things. Either there is no mitochondria or there is not enough oxygen, right? Yes. If mitochondria is not functional and oxygen is not enough, there is lactate formation. Yes or no? Are you clear with that? Now, what is the problem in cancer cells? Cancer cells is their lack of oxygen initially, not later. They have their own angiogenesis. So there is enough oxygen. Then why there is lactate? Because what is defective in them? No, cancer cells initially they have low oxygen, but then they start having their own angiogenesis. So they have oxygen to survive. But what is wrong with them? Mitochondria. So do you know what is Warburg effect now? Warburg effect is forming lactate in the presence of enough oxygen. What is Warburg effect? Forming lactate in the presence of enough oxygen. But why are you forming? Because what is wrong? Why lactate is formed? Not because of oxygen, but because of lack of functional what? Mitochondria. Now, very important for you to understand that when mitochondria is not there, so you understood this point? Why there's lactate formation in cancer cells? Not because of oxygen, lack of oxygen. They have enough oxygen because of their own angiogenesis, because of lack of mitochondria. But please understand that lactate formation promotes more and more mutation and cell survival. Why? Because that forms a soil for more mutation and more survival of the cells. Secondly, remember, are mitochondria defective in cancer cells? Are mitochondria defective in cancer cells? Yes. Mitochondria are also involved in apoptosis. So when mitochondria is defective, you lose apoptosis. Is it good for cancer cells? Is it good for cancer cells to lose apoptosis? Yes, because then cancer cells can survive. Yes, it's good for cancer cells. That's why they evade apoptosis. So that happens because of defective mitochondria too, because mitochondria is involved in apoptosis. So do you understand this Warburg effect, where you form lactate in the presence of enough oxygen is low or mitochondria is defective? Why are you forming lactate here? In case of Warburg effect, the mitochondria is defective. But there are other repercussions of that too. Because mitochondria is defective, are you forming lactate? Lactate is a good soil for more mutation, more cell survival of these mutated cells. Secondly, mitochondria is also involved in apoptosis. So you will lose apoptosis, and that's favorable for cancer cells. It's not favorable for the body, but it's favorable for understand that concept. Evading apoptosis is not favorable for our body, but is favorable for to evade apoptosis is favorable for which cells? Not the normal cells, but the cancer cells. So do you understand that the lactate formation is because of mitochondria defective, not oxygen? Lactate formation promotes more mutation and cell survival of these mutated cells. And what is PAT scan? Who will tell me what's PAT scan? How can it help you detect tumors? Because are cancer cells rapidly dividing cells? Are they rapidly dividing cells? So what do they do? What can they quickly use for their energy? Glucose. When they can use for glucose, will they do aerobic glycolysis or anaerobic glycolysis? Will they do aerobic or anaerobic glycolysis, cancer cells? What can they usually quickly, because they are proliferating so fast, what do? They will pick up glucose for their needs, but they will do anaerobic glycolysis. And that is why there is more lactate formation. So you understood that? And why there is anaerobic glycolysis? Because of oxygen problem or mitochondria? Yes, they are fast. Fast is making them use of glucose, not fat and uh, not lipid and ketones, because they need quick energy, right? Rapidly dividing makes them use glucose. That's why tumor cells take up a lot of glucose. But what makes them lean towards lactate formation? Is it oxygen or mitochondria being defective? Mitochondria being defective. So do you understand that? Yes. So what's PAT scan? Wherever the cells are taking up too much glucose, there could be, you could detect tumors because of that. What do you give in PAT scan? Fluorodeoxyglucose, that's an analog of glucose, and that can be detected. And more uptake means there is cancer. So there is a tumor that would have increased uptake of glucose. So you understand why PAT scan is dependent on that rapid uptake of glucose. So you should know normal or altered and then PAT scan, right? Is there any confusion? PAT scan is not just evaluating metabolic activity of the cells. PAT scan is simply evaluating the rapid uptake of glucose by tumor cells. That's all. There's a rapid uptake by tumor cells because they are rapidly proliferating and they lean towards anaerobic glycolysis, forming what? Forming what? Anaerobic means they will form what? What will be the product? Lactate. Lactate is another bad soil for more mutation, more survival of these defected or mutated cells. And then mitochondria is defective. So these mutated cell also evade what? Since mitochondria is defective, these mutated cells also evade apoptosis, right? So are you clear with that? So mitochondria are functional. That's not true, right? Is oxidative phosphorylation defective in them? Is oxidative phosphorylation defective in them? Because of mitochondria, you can say yes, yeah, there is no oxidative phosphorylation. Is it helping? Are you clear with the concepts now? Great, let's move forward. So you know was this Warburg effect. Lactate formation in the presence of enough oxygen. Reason is mitochondria is defective. You need to understand there are only two ways lactate can be formed: mitochondria defective or oxygen non-presence. In case of cancer cells, that's why there are two theories. Don't waste your time on that. Some people lean on nuclear theory, some lean on mitochondrial theory. I think you should combine them both. What's the nuclear theory? Gain of function of. What is the nuclear theory? Gain of function of proto-oncogene and loss of function of tumor suppressor gene. What's mitochondrial theory? Mitochondria are defective. Mitochondria are involved in apoptosis. Cancer cells evade apoptosis. Does that make sense? Yes? And are you following my pace and is this helping? Great. Now let's look at this. Now you should not be confused with this. It is either or. It's not that you need both for cancer. Either gain of function of proto-oncogene or loss of function of tumor suppressor gene. So which choice is giving you that answer? Only choice C. It's either or. Now, is this even correct? Now we will go to what is the mechanism. Do you need two gain of function mutation for proto-oncogene? No, you just need one gain of function mutation for proto-oncogene. But do you need to knock out both the tumor suppressor gene? Do you need to knock out both the tumor suppressor gene? I would say allele, please, because each gene has two alleles, right? So you would need only one gain of function mutation allele for proto-oncogene, but you need to knock out both the allele for tumor suppressor gene. because each gene has two alleles, right? Does that make sense? Now, please, students are confused with this. To me, tell me, this looks like a mechanism of tumor suppressor, look like mechanism for autosomal recessive, and proto-oncogene looks like a mechanism of autosomal dominant, right? In autosomal dominant, you need only one gene, but that's not true in cancer cells. Cancer cell is not like our typical inheritance pattern genetics. Cancer cells, whether all inherited cancer cells are autosomal dominant. It's not based on the mechanism here. It's based on presentation. I will tell you why. So whether it's gain of function mutation of proto-oncogene 1 and loss of function mutation of tumor suppressor gene, both alleles, still both proto-oncogene, tumor suppressor gene inheritance pattern is what? What is the inheritance pattern for both? Autosomal dominant or recessive? Whether proto-oncogene tumor suppressor inherited syndromes are autosomal dominant or recessive? Dominant, please know that. Secondly, students are confused in what is inherited and what is sporadic. I will tell you. But in inherited syndrome, you will inherit the mutation. In sporadic, the chance is totally sporadic and there is no inherited mutation. I will tell you how they both are different. Not now when I come to the slide. So you should know proto-oncogene, tumor suppressor gene. You should know one gain of function and two loss of function for tumor suppressor gene. You should know both are autosomal dominant if inherited. You should also know what is the difference between inherited and sporadic, right? But don't go into details of that. At least try to identify the terms. Are you clear with them now, right? So this is not even correct. So that is why either or. Are you clear with that? Is there any confusion, right? Let's go further. Now you should know, please, every gene, I'm telling you how you will do this section, gene, function, syndrome, presentation. You got to fill this line. Gene, all genes are always in caps. Gene, function, function you should know, please. Cancer syndrome, because they're all inherited, and presentation. I will tell you presentation is only by tumor, whether it's lung, breast, endometrial, colon, that's presentation, not do all the presentation. Please don't go to ChatGPT for that, right? You're not being tested on presentation. But I want you to know, for example, tell me the tumor of retina. Which one am I talking about? Tumor of retina, which inherited syndrome I'm talking about? White eye reflex because of the tumor of the retina. Retinoblastoma, I'm not talking about Leaf-Ramini syndrome. Leaf-Ramini syndrome, what is common? Sarcomas. Sarcomas are very rare. But in Leaf-Ramini syndrome, sarcomas are common. If you see sarcoma, buzzword is Leaf-Ramini. But if you see tumor of retina, retinoblastoma. So I will tell you similarly. So let's finish that cascade. What? Gene, function I will not tell you now because it will come up. I will only tell you gene, clinical syndrome, and presentation. First of all, in blue box is tumor suppressor gene. In green box is proto-oncogene. What are these? Receptor tyrosine kinase. Please note down. You should know the difference between receptor tyrosine kinase and intracellular tyrosine kinase. I will bring that. You should know the difference between receptor tyrosine kinase and intracellular tyrosine kinase. Receptor tyrosine kinase are used by which signaling? Can you give me a signaling that use receptor tyrosine kinase? Give me the signaling molecule. Insulin, but we are not talking about insulin here. Anything else? Anything else that uses receptor tyrosine kinase? Insulin is not involved in cancer here. Please give me an answer. Growth factor. Very good. Growth factor uses receptor tyrosine kinase, but receptor tyrosine kinase of receptor activity is different from intracellular tyrosine kinase. Please note down that first. Number one, these are all receptor tyrosine kinase. In green is proto-oncogene. You don't need to remember the tumors, but they are proto-oncogenes. You need to remember the gene, the inherited syndrome, function for tumor suppressor gene. Absolutely. So now look at this. What is this? Gain of function of green box or loss of function will lead to cancer? MET and RET, they are receptor tyrosine kinase. Gain of function, so don't flip-flop that. For green box, it's gain of function. For blue box, it's loss of function. Now let's do clinical presentation. Retinoblastoma, you will see tumor of the retina and white eye reflex. When you shine a light on the eye, it looks white, not yellow. So white eye reflexes retinoblastoma. BRCA1, BRCA2. I will finish the function here only because this you have not learned in spring, you have learned in fall. Tell me, where was BRCA1, BRCA2 involved? Where was it involved? Single-stranded, double-stranded? BRCA1, BRCA2. BRCA1, BRCA2. So you should know gene, function, clinical syndrome, and presentation. I'm finishing some of the functions here which you learned in fall, not spring. They are involved in double-stranded homologous repair. BRCA1, BRCA2 are involved in double-stranded homologous repair, and that will be lost. But please, please, and please, it is breast and ovarian, not endometrial cancer. Endometrial cancer is not BRCA1, BRCA2. If you see breast and ovarian, it is BRCA1, BRCA2. If you see endometrial, I will tell you where, but this is not endometrial, right? BRCA2 is also breast and male. BRCA2 is breast and male, but breast and ovarian, not endometrial. You know this is colon cancer, right? APC, presentation colon cancer. Leaf Ramani, presentation sarcomas. Sarcomas are very rare, but common in Leaf Ramani syndrome. What was this? You remember? You did that in repair too. So retinoblastoma, tumor of the retina, white eye reflex. Breast and ovarian, not endometrial, BRCA1, BRCA2. Colon cancer, APC. If you see colon cancer presentation, go to APC. If you see tumor of the retina, go to RB. You understand where I'm taking you to? Presentation, sarcomas, you will go to P53. Yes? What about MLH, MSH? MLH13, MSH26. You don't need to know the number. Remember MLH and MSH are in? MLH and MSH were what? Were they, what are MLH? MLH, MSH. They are mismatch repair genes. Mismatch repair genes. Don't forget that. MLH and MSH misrepair gene, Lynch syndrome. What will you see in Lynch syndrome? Tell me, which cancers? Lynch syndrome, also known as human non-polyposis colon cancer. HNPCC or Lynch syndrome. Which cancer you will see? HNPCC, Lynch syndrome. It's just like FAP, colorectal cancer, but now in female, you see endometrial cancer. In female, here you see endometrial cancer. So if you see endometrial cancer, that's not BRCA1, BRCA2. If endometrial cancer, along with colorectal cancer, what is it? Endometrial and colorectal is what? Mismatch repair genes, right? So are you clear with that? Neurofibromatosis, there are neurocutaneous tumor, you know that. I will tell you the function of RB, very important. I've told you the function of BRCA1, BRCA2. I will tell you the function of APC, P53. Have I told you the function of MLH, MSH? They are involved in mismatch repair. NF1, I will tell you, I've not told you. And these are what? Receptor tyrosine kinase. Who uses them? Growth factor, right? Are you clear with this slide, right? So please do in blue and their clinical syndrome. Please, please do each one of them. Not in detail. I told you gene, its normal function, clinical syndrome, and key primary presentation I have given for all. Are you confused still in any one of them? Is there any confusion in clinical presentation? Great. Is this helping? I'm going a little bit slow, not too slow also, right? That's because it's a very complex content. I don't want to rush over it. Once you're done with this, you're good. Now, did I tell you? I'll tell you what are tumor suppressor genes, caretaker, and gatekeepers. So please know them, first of all. Very good. I told you that. For a proto-oncogene, please, please, and please, first you should know what are your normal genes. Then you do that. I told you, first gene, their function, please As I always tell you, don't jump to clinical. First, you should know normal to know abnormal. What are proto-oncogene? You don't have to memorize this. Look at this. Growth, you have done that. Growth factor receptor. What are they? What activity they have? Growth factor receptors. What activity they have? Receptor, tyrosine kinase. What example I will give you? HER1, HER2. HER1 is lung cancer. HER2 is breast cancer. So growth first, normal. Growth factor, I'll go to abnormal later. Growth factor receptors. Then that RAS, RAF, MAP kinase pathway. Are you aware of that? This is ringing sound. RAS, RAF, MAP kinase pathway. When is RAS active? When bound to? When is RAS active? Very, very important. Cannot miss that. RAS is active and bound to. Give me the answer. RAS is active and bound to? Bound to? GDP or GTP? GTP. Please don't flip-flop that. Note it down. RAS is active and bound to GTP. But in the end, there is MYC. Don't forget MYC gene, please. MYC is the end of the growth factor signaling pathway. It's a transcription factor that allows cell proliferation. So again, growth factor receptors, RAS, RAF, MAP kinase pathway, and then there is MYC. MYC in the end is known as transcription factor. It helps in cell proliferation. Now tell me, any gain of function of growth factor receptors, RAS, RAF, MAP kinase, MYC, what will that lead to? Should it be gain of function or loss of function? All of them. The whole growth factor signaling, gain of function that will lead to cancer. Please know that. Please know that, right? So growth factor signaling is gone. I will finish the cascade of MYC. You should know that, but at least I have told you up to MYC. What does MYC do? I will tell you later. Anything in growth factor signaling pathway. Also, I'm sorry, I was thinking, what am I forgetting? Receptor tyrosine kinase is different from intracellular tyrosine kinase. All these RAF, MAP kinase, they are intracellular tyrosine kinase. In the end, the receptor tyrosine kinase is at the top of the receptor. Now, what are tumor suppressor genes? Gatekeeper and caretaker. As the name suggests, what will they take care of? What will the caretaker take care of? Any DNA damage or, for example, mismatch repair genes, BRCA1 and BRCA2. What are these? Gatekeeper. Please, you should know function of each. What is the function of mismatch repair? They will repair mismatches. What is the function of BRCA1, BRCA2? They are involved in, you should know function. The BRCA1, BRCA2 are involved in double-stranded homologous repair. Very good. I will tell the function of others. Now, how do you, this is the we are just at the key. How do you identify? Now, what are they saying first? First, what are they asking? Is it gain of function or loss of function? So it should be for proto-oncogene or tumor suppressor gene. Proto-oncogene. So anywhere there is tumor suppressor in the answer, don't even see that. Is this tumor suppressor? A. RB, is it tumor suppressor? So it's not a correct answer. So which is the correct answer? Anywhere there is no tumor suppressor gene, which is RAS. And what is RAS? RAS is always active and bound to? Is RAS on the receptor or intracellular? Is RAS intracellular? Yes, it's always bound to the GTP. And this is growth factor receptors, epidermal growth factor receptor. What activity it has? This is receptor tyrosine kinase, right? So what is your answer? Should be anywhere there is a tumor suppressor gene, you will not pick that. Why? Because what are they asking? What are they asking? Are they asking loss of function or gain of function? Gain of function. So are you clear with that? Let's go further. So far, everything is good. So are you aware of gain of function? Sorry, gain of function for proto-oncogene and loss of function for tumor suppressor gene? Any confusion? Both are autosomal dominant, but you need one gain of function allele and for proto-oncogene and two loss of function for tumor suppressor gene. Please, anything in growth factor signaling, what's the last transcription factor here in growth factor signaling right at the bottom? MYC, please know MYC gene very well. Anywhere MYC is in the lecture, Please do those slides, very important. Have we done this? HER1, now pathology. They are a receptor tyrosine kinase. HER1, lung cancer. HER2, breast cancer. RAS, when bound to GTP, also in some of the cancers. What was this? BCR, Abelson. You should know chromosomal translocation very well. I'll give you quickly at the end. BCR, Abelson gene. Which chromosomal translocation? Where is it seen? AML, PML, or CML? AML, PML, or CML? Where is CML? Chronic myeloid leukemia, BCR, Abelson fusion gene known as Philadelphia chromosome. Now here also comes MYC. So MYC is different in different disorders, please. Keep an eye on that. I'll explain you why. But is MYC involved in this? Is MYC in CML? Is MYC gene in CML? No. Did I say it's in CML? I didn't say that, right? I was asking you, sorry. Yes, MYC is not in CML. Where is MYC then? In CML, it's an intracellular tyrosine kinase becomes active. Where is MYC? Which chromosomal translocation is MYC? It's not in CML. Which chromosomal translocation is MYC? Burkitt's lymphoma or follicular lymphoma? Burkitt's or follicular? Burkitt's or follicular? CML, there's no MYC. No, wherever MYC is Burkitt's. Don't forget that. Burkitt's lymphoma, it's MYC, I'll tell you. Right? So do you understand proto-oncogenes, gain of function, loss of function, tumor suppressor gene? Are you aware of gatekeepers and caretakers? Now look at this, very, very important. This is students tend to forget. It's not just the mutated cell, they have to survive. So what should they evade? What do cancer cells evade? What is favorable to cancer cells? To evade? Evade what? Apoptosis, right? So which one should be, they want to evade apoptosis. So which one is tumor suppressor and which one is proto-oncogene? Please note apoptotic is FAS, anti-apoptotic is BCL2. So which one should be tumor suppressor and which one should be proto-oncogenes? Which one they want to lose and which gene they want to gain? Which gene they want to gain? They want to gain FAS? They want to undergo apoptosis, cancer cells. What is favorable to cancer cells? To gain FAS or to gain BCL2? To gain BCL2. So BCL2 is a proto-oncogene and FAS is a tumor suppressor gene. That's why under proto-oncogene is given BCL2. Please know that. Gain of function is for apoptotic or anti-apoptotic? Gain of function. Apoptotic or anti-apoptotic? Gain of function. Gain of anti-apoptotic is a proto-oncogene. They want to gain BCL2 and they want to lose FAS. Please, please don't mess up this concept. Very, very important. I know tumor is a little bit tough, but I think you want me to finish it all right? Quickly, right? Or you want a 5 minutes break in between? You want to finish it? Great. Here comes, keep going, keep going. Yeah, I'm keep going since fall. So without a break. So look at MYC gene. I told you no MYC gene. Now MYC is a proto-oncogene or a tumor suppressor gene. Did I tell you growth factor receptor, RAS, RAF, MAP kinase, and MYC? They are all what? They are proto-oncogenes. They're all proto-oncogenes. So then what is the mutation? Gain of function or loss of function for proto-oncogenes? Gain of function or loss? Gain of function. Now look at this. Just a second. Termination, disruption, loss of heterozygosity, and hypermethylation. Are they gain of function mutation mechanism or loss of function? Termination, disruption, loss of heterozygosity, and hypermethylation. Are they look like gain of function mechanism or loss of function? They are all loss of function. So will you even look at B, C, D, and E? So that's why it's important for you to know what is the gene and what is its mutation that will lead to cancer. So MYC is gain of function or loss of function? Gain of function. So B, C, D, and E are lame choices. There's only one choice here, which is A, and that should be the right answer. Does that make sense? Please rule out your answer. Don't memorize them. But now, so MIC is gain of function. But now I will explain you that these two are different mechanisms. Though MIC is not loss of function, yeah? Are you clear with that? But there is difference in MIC amplification versus MIC overexpression. MIC amplification, MIC overexpression is different. And a hyperactive protein is also different. I will show you that. They are different mechanism. But look at this. What will this do? A point mutation in the coding region. Will it cause overexpression? Is this overexpression amplification, or this can lead to a hyperactive protein? Remember, hyperactive protein is not normal. So this mutation in the coding region, will it lead to a hyperactive protein or an overexpressed protein? Let's only do two. Tell me. This mutation, so gain, you know MIC is gain of function. You're clear with that. Are you clear that B, C, D, and E should not even be the choices? Yes? Now tell me how this will lead to gain of function mutation. This mutation will lead to a hyperactive protein or overexpressed? Not overexpressed, hyperactive protein. I will tell you how. There comes these two mechanisms of gain of function, amplification and overexpression. They are not the same. First of all, don't miss them where they are. They are not same mechanism, but they are both gain of function. Are they both gain of function, first of all, mechanism? Are they both gain of function mechanism, but they are different. So when you say MIC amplification, they are talking about neuroblastoma and MIC. It is the third common tumor in children. It's an extracranial tumor in the extracranial tumor, not brain tumor. First in children is most common cancer is leukemia, then there is brain tumor, then is neuroblastoma. Neuroblastoma is not brain tumor, it's an extracranial tumor. But when they talk about MIC amplification, they are talking about neuroblastoma. But when they say MIC overexpression, that is Burkitt's. So also be mindful. If you see MIC, you can jump to neuroblastoma. Don't do that. They should say MIC amplification. If you want to pick Burkitt's, they should say MIC overexpression. So let's see how they are different. So first of all, you didn't answer me that correctly, the first one. A point mutation in a coding region, look at this. A point mutation in the coding region is leading to what? Is it leading to overexpression or hyperactive protein? Please, you should know 1, 2, 3, 4 so that you don't make a mistake. Yes. So yes, point mutation could be a mechanism of proto-oncogene, but that doesn't lead to overexpression. What do you mean by overexpression here? The protein is wild, but it's overexpressed. Again, cancer. Here, the protein is not wild. There's a hyperactive protein. For example, tyrosine kinase domain is turned on all the time. Is it a hyperactive tyrosine kinase? For example, the tyrosine kinase domain is turned on all the time. Is it a hyperactive tyrosine kinase? Yes? Is it a hyperactive tyrosine kinase? That will lead to cancer. But look at the next one. Next is when promoter is mutated and there is overexpressed protein. Now, this is wild protein, but formed in large quantity. So are they two same? Are they two same? No, this is a hyperactive protein. This is not a wild protein. This is a wild protein overexpressed. And look at this gene amplification. Too many copies. Here, promoter is affected. Here, promoter is not affected. Here, gene is amplified. But still there is what? Overexpression. Is this also a wild protein? So both are example of wild protein, but overexpressed. In this case, promoter is mutated. In this case, gene is amplified. And gene amplification is for which? And MIC amplification is which one? Neuroblastoma. So we understand that there's the same mechanism of proto-oncogene. There is gain of function of proto-oncogene, but different mechanism. Now, what is this? This you should know chromosomal translocation. So are you clear with this? Which was answer A, hyperactive protein. For example, tyrosine kinase turned on all the time. Yes. Promoter can be mutated or gene can be amplified. But here, protein is normal, overexpressed, just overexpressed. Are you clear with that? Yes. Where will you see MYC amplification? Neuroblastoma. Where will you see MYC overexpressed? Not amplified, but overexpressed. So MYC amplification is not Burkitt's lymphoma. Burkitt lymphoma is MYC is overexpressed. I will tell you how. Now look at this one. Chromosomal translocation results in two things, please. Either the gene is under the influence of strong promoter after translocation. This is the strong promoter from another gene. So what there are two results of chromosomal translocation. Either the wild gene is under a strong promoter. What will happen if a wild gene comes under a strong promoter from another gene? What will be the result? If a wild gene is under a strong promoter, what will be the overexpressed? So is this overexpressed? Is this overexpressed? And is this overexpressed? Are three the same? But the mechanism is different. Here, promoter is mutated. Here, gene is amplified. Here, there is translocation. A wild gene is under a strong promoter. What is the example of these chromosomal translocation? Burkitt's lymphoma and follicular. Don't forget that. What are the example under these two? Where the wild gene comes under strong promoter? Burkitt's lymphoma and follicular lymphoma. What is the next? Look at this. Two exons have fused. This is a gene fused with another gene, and their product is oncogenic. That's known as fusion gene or chimeric gene. After translocation, this happens. These are both outcome of translocation. What are the example under fusion gene? CML and acute promyelocytic leukemia. PML. AML or PML and CML. So what is wild gene under strong promoter after translocation? What are example under these? Where a strong promoter comes in, a wild gene comes under the influence of a strong promoter after translocation, Burkitt's and one more, Burkitt's and the follicular. And what is fusion gene example? When two exon fused, AML or PML, one more, CML. So are you clear with that? Very good. Very good. Don't flip-flop them, please. I explain you. This is a very good slide to understand everything, right? I'm reinforcing things. I've explained in video, but video has very limited time. That's why interactive session help, right? Again, is there loss of heterozygosity? I will tell you what's loss of heterozygosity. So loss of heterozygosity is for proto-oncogene or tumor suppressor gene. Proto-oncogene or tumor suppressor gene. Loss of heterozygosity is for proto-oncogene or tumor suppressor gene. Tumor suppressor gene. So what should be your answer? A. Now tell me why C is not the right answer. Anti-apoptotic gene is proto-oncogene or tumor suppressor gene? Anti-apoptotic gene is a proto-oncogene. And what are they saying? Loss of heterozygosity. So what should have, this should have been the answer when they ask you gain of function. So you gain you gain anti-apoptotic. And what is favorable for cancer? Not, I don't think I should say you gain. What is favorable for cancer cells? To gain anti-apoptotic, which is BCL2, and lose apoptotic, which is which one? Apoptotic is which one? FAS. Don't forget that. So should C be the right answer? Should C be the right answer? When they're asking loss of heterozygosity. No. You want, cancer cells want to gain them, not lose them. So are you clear with these concepts? You are going almost to the end. I want to explain you a few more concepts. I have stressed on this. Are you aware of gatekeepers and caretakers? What are they? Tumor suppressor or proto-oncogenes? What are they? Tumor suppressor or proto-oncogenes? Gatekeeper, caretakers. Gatekeeper, tumor suppressor gene. Please, everything in green is proto-oncogene. Gain of function mutation of growth factor receptors, RAS, RAF, MYC. They are all gain of function mutation genes. Everything in green is gain of function mutation because they are growth factor signaling pathway. I want to complete this pathway by giving you function of P53 and RB. You should know function of P53 as well as RB. Let's do first function of P53. Both are phosphoprotein, active and phosphorylated. Who phosphorylates P53? Tell me. Who phosphorylates P53? What will phosphorylate P53 in your body? DNA damage. P53 is always produced inside your cells, but whenever there is a DNA damage, P53 is phosphorylated. What does it transcribe? Look at the gene and protein. What does it transcribe? P21. What is P21 doing? Stopping. What is P21 doing? Stopping what? G1S phase or cell cycle. Why? Tell me why. Whenever there is a DNA damage, what happens to P53? It's phosphorylated. What does it transcribe? P21, and it stops the cell cycle. Why? Why does it stop this? Why P53 stops the cell cycle? Yes, to prevent the DNA damaged cells to go into the cell cycle. So let's see, there are three functions of P53. First of all, whenever there is a DNA damage, it becomes active. It transcribes P21 and stops cell cycles. This is first function of P53. Why does it stop cell cycle? To allow DNA for repair. If repair happens, P53 goes down. But if repair does not happen, what should P53 induce, which is the third function of P53? Apoptosis. Very good. So what is this is normal. Normal function of P53 is activated by, always produced in the cells, but activated when there is DNA damage, transcribes P21. What does P21 do? First foremost thing is stop the cell cycle. Second thing, allow DNA to repair. So will induce DNA repair enzyme. And lastly, if DNA repair did not happen, apoptosis of the cells. Now imagine what is favorable for cancer, to gain P53 or lose P53. P53 is a brake. It's a cell checkpoint. It's a gatekeeper to lose P53. Have you understood the function of P53? Now let's see RB. RB is also a tumor suppressor gene. Now, what is cyclin D? What is MYC doing? MYC is a transcription factor that will allow cell progression or cell proliferation. MYC transcribes cyclin CDK4. They are kinase and they phosphorylate RB. So what is the function of MYC? Is MYC at the end of growth factor signaling? Is MYC at the end of growth factor signaling? What does MYC transcribe? Cyclin CDK4, which is a kinase, which phosphorylate RB. Now, this is very important for you. When RB is phosphorylated, E2F is released. When E2F is released, what else is transcribed? What else is activated? Sorry. When RB is phosphorylated, what is released? E2F, and E2F activates cyclin E CDK2 complex, which causes cell proliferation. So let's do the cascade again. Whenever there is a growth factor signaling, will MYC be produced? Whenever cell has growth factor signaling, will MYC be produced? Will you have MYC? What will MYC activate? Cyclin CDK4. That will phosphorylate what? CDK4 will phosphorylate what? Which RB. When RB is phosphorylated, what will be released? E2F. E2F will activate cyclin E, CDK4 or 2? 2, and that will start cell progression and cell proliferation. So what is favorable to cancer cells? To gain RB or to lose RB? To lose RB. Now, though this is a big cascade, I'll give you a small mnemonic. RB, function of RB is to sequester E2F. Please know that. Function of RB is to sequester E2F. When will RB release E2F? When there's a growth factor signaling and when there is no growth factor signaling. Function of RB is to sequester E2F. RB will only release E2F when there is when the cell has what? Growth factor signaling. So in a quiet cell, what is RB doing? In a quiet cell where there is no growth factor signaling, what is RB doing? RB is sequestering what? E2F. Just know that. Is that clear? Are you clear with function of RB? Yes? So let's go further. Now this is loss of function. So which one is loss of function? Which is the tumor suppressor gene? FAS or BCL2? FAS or BCL2? Which is FAS? favorable of cancer is to lose apoptotic gain and anti-apoptotic genes. But are you aware of tumor suppressor gene, gatekeeper and caretaker? Are you aware of function of P53, RB? So tell me one, if you have understood RB, tell me when is RB phosphorylated? Who phosphorylates RB? Growth factor signaling or no growth factor signaling? When is RB phosphorylated? When there is growth factor signaling or when there is no growth factor signaling? When there is growth factor signaling. Very good. So no MYC, no P53, no RB. Now, loss of function mutation is for a tumor suppressor gene. Tell me, is there is only knockout of one allele? Is this cell normal or cancerous? Is this cell normal or cancerous? This cell, which I have circled, is it normal or cancerous? It is absolutely normal. You have to knock out both the alleles of the same gene in the same cell to lose the tumor suppressor function. For example, if loss of function was, if an individual has a loss of function allele for BRCA1 and he inherits a loss of function allele for BRCA2, will there be cancer? He has one loss of function of BRCA1 and the other loss of function for BRCA2. Will these two cells be cancerous? No, until there is a second hit in BRCA1 and a second hit in BRCA2. So if you inherit one loss of function of BRCA1 and one loss of function of BRCA2, still no cancer, no breast and ovarian cancer. In BRCA1, BRCA2, you have to knock out the other allele, which is this one. Or I can ask you another way. If someone inherits a loss of function allele for BRCA1, will that be cancerous? Just one loss of function allele for BRCA1. Will that be cancerous? No, until the second hit. So first hit in inherited cancer is inherited, and second hit is somatic and acquired. In somatic cells, acquired is by chance. This is by chance. That's why people ask me that if you inherit a tumor suppressor allele, loss of function, why there is no cancer? Because you still have to knock out the other allele, and that is by chance. That is by chance and acquired. So that is why there is no cancer until you knock out both the alleles. Is that clear to you? Yes. The other thing is it should be in the same gene and same cell. So for example, Leaf-Ramani syndrome, I told you there are sarcomas. Yes, sarcomas are very common. So where did the second hit happen? Which cells? Sarcoma is from which cells? So how do you know why only sarcoma? Why not breast, ovarian? So why in Leaf-Ramani syndrome, the sarcoma, there are sarcomas because sarcoma is for which tissue? Epithelial or connective tissue? Sarcomas. Is that tumor of epithelial cells or connective tissue? Connective tissue. Epithelial is carcinoma. Sarcomas are connective tissue. So why in Leaf-Ramani are there only sarcomas? Because second hit happened in the connective tissue cells. So that's why there is sarcomas. It should be in the same, it should be the second allele of the same gene and the cell too. Wherever there is a second hit, they get the cancer, right? Does that make sense? The second hit for sarcoma should be in the connective tissue cells. Now, what about epigenetic modification? Tell me, methylation of a DNA containing a tumor suppressor gene or deacetylation of histone region containing tumor suppressor gene, will there be cancer? Methylation of DNA of the region that contain tumor suppressor gene or deacetylation. I'm not talking methylation in histones, please. Methylation of DNA and deacetylation of histone regions that contain tumor suppressor gene. Will there be cancer? Yes, because both will lead to loss of function. So there are epigenetic functions also, because remember, will methylation of DNA and deacetylation of histone a mechanism of proto-oncogene? Will methylation of DNA and deacetylation of histone a mechanism for proto-oncogene? Methylation of DNA and deacetylation of histone, will that be a mechanism for a proto-oncogene? No, because that's loss of function. That is silencing. Loss of function silencing is for tumor suppressor gene. Proto-oncogene is gain of function. So are you getting the concept? I want you to know the concept. Please, since miRNA was not answered well in the last exam, I still want to tell you only one thing. miRNA do what? Do they degrade mRNA? I'm just asking you still. I think you don't need to know this. This is very advanced, but I still want to ask you so you understood so much blabbering that I did, right? miRNA degrade mRNA? Do miRNA degrade mRNA? Yes or no? And protein expression is low. Do miRNA degrade mRNA and protein expression is reduced? Yes. Now, oncomeres are the miRNA that target tumor suppressor gene. Listen to me carefully, because the exams right now, because we have only few questions to test. So we don't test first order questions. We test second and third order questions. So listen to me carefully. You know the concept, miRNA degrade mRNA and reduce protein expression. Now, oncomeres are the miRNA that target tumor suppressor gene. Oncomeres are the miRNA that target tumor suppressor gene. Tell me, activation or inactivation of them will lead to cancer. Activation or inactivation of activation. Somebody who's saying inactivation is losing the concept. Remember, miRNA target with genes? With genes. What did I tell you? These oncomeres, they miRNA that target with genes. Which genes did I tell you they target? Tumor suppressor. So you want their activation to lose them, right? Not inactivation, right? You want the activation to lose them, right? Just to understand the concept. Second hit is important, please. With one hit, one allele, you don't get cancer. Inherited cancer syndrome are autosomal dominant. Why? Because you inherit one knock of function, and to have a second knock is very common. It's good. It's not rare. It's common to get a second knock. That is why inherited cancer syndromes are autosomal dominant. They have multiple primary tumor and occur early in life because they inherit one knock function and they have to just knock out the other, which is acquired and more common. That's why they are autosomal dominant, right? In sporadic cancer, do you acquire any loss of function allele in sporadics? In sporadic cancer, do you acquire any, do you inherit any loss of function allele? No. Both the knockout are by chance, sporadic, and that's very rare. That's why sporadic cancers do not have multiple tumor and do not occur early in life. I don't want you to get lost in it. Just know that what is loss of heterozygosity? Tell me what's loss of heterozygosity. Loss of heterozygosity means here one is wild and one is mutant. Red is wild and green is mutant. Yes. Loss of heterozygosity means you lose the normal also, convert it into mutant. Then both will be mutant. Does that make sense? Right now it's heterozygous. One is wild and one is mutant. Yes. If you convert the wild into mutant also, there's loss of heterozygosity. That's why it's known as, that's why you have to knock out both the alleles, right? To lose the tumor suppressor function. All right. This is very simple. I think I only have 5 minutes. You want to take a break or you want me to finish it? You want me to finish this? Yes? Everyone? Can I take a 5-minute break? I need it. I think there are only 5 slides left. So is it okay, everyone? Just 5 minutes, please. I'm here. We are at the end. Thank you. Maybe we can take a quick break and come back when everybody's back. I needed a little sip of water. I mean, I'm blabbering a lot today, but I hope this is helping. Maybe I'll wait for everybody to come back. And great, I'm glad. Great, yes. I think when we will come back, we will have more time for questions. Let me only finish. Just I want to redo this section if anything was unanswered, right? I will wait for everyone to come back, so don't panic. When everybody's back, just give me a heads up, please. Yeah. Everybody's back. Who wants to give me a heads up? Who's taking the heads up? Okay. If you can, unpause your video, I would know you're back, but
Yes,
ready?
everybody's back for the
Okay.
cameras that were on prior. Yes.
Okay, great. I just don't want anyone to miss it, right? Because I said a short break. I didn't say how short it is. So, retinoblastoma. Now, retinoblastoma, please don't get confused. Though the normal function is, now that confuses students, which I'll ask you now. Yes, normal function is at the bottom, right? Sorry, let's not do the answer. Normal function is whenever there's growth factor signaling, will RB be phosphorylated? Whenever there's a growth factor, RB, what will it release? What will it release? E2F, and then there is cell progression. No cyclin CDK4 where it is, cyclin E CDK2 where it is. I'm not fitting in here, I've told you. What happens to P53 whenever there is DNA damage? Is P53 phosphorylated also? And what does it transcribe? P21 and stops the cell cycle. Though there are three outcomes, P53 will stop the cell cycle, allow DNA to repair. If DNA repair did not happen, will induce apoptosis. But look at here, in retinoblastoma, there is no RB. It doesn't matter, it's phosphorylated, dephosphorylated, doesn't matter. There's no RB. You lost the function, you had two knockout of allele, so there's no RB. So what will happen? What will happen to E2F? It's not about phosphorylated, nothing. What will happen to E2F when there is no RB? E2F will always be? E2 will be? No, not remain sequestered. Now let's do that. When the cell is quiet, what does RB do? When the cell is quiet, what does RB do? Release or sequester E2F when cell is quiet. What does RB do to sequester E2F? When there is growth factor signaling, RB releases E2F. But right now there's no RB. What will happen to E2F? There's no RB function. What will happen to E2F? Tell me. No. Always released. RB was only sequestering E2F, right? Who was sequestering E2F? Who was allowing E2F not to be released? RB. RB sequestered E2F. When there was growth factor signaling, RB released E2F. Now there's no RB. So what will happen to E2F? There's no phosphorylation, there's no dephosphorylation, there's no question about that. Because there's no RB function, E2F will always be released. And that leads to cell cycle progression, G1S, and cancer. Do you understand that? Here students get confused. Oh, RB will be phosphorylated, hypophosphorylated. That is normal. RB, when phosphorylated, releases E2F. But here, RB function is lost. So E2F will always be released. You should understand that RB sequesters E2F when there is no growth factor signaling. RB will only release E2F when there is growth factor signaling. Because when do you want growth? Only when there is growth factor signaling. Does that make sense? Otherwise, it will be cancer. Oh, RB is captive, so No, not RB is not captive. RB has lost its function. So now RB is no more sequestering E2F. That is why the loss of function E2F will always be released. Right? Are you clear with that? Retinoblastoma, because there's a tumor at the back of the eye, there is, have I mentioned white eye reflex? Yeah, sorry, I mentioned here. Because there's a tumor at the back of the retina, there is a white eye reflex, which the physician also does. The parents can also note a white eye reflex because of the blocking the retina. Right? Again, see here to understand RB. What is this? Is the growth factor signaling on the left? Is growth factor signaling on the left? Because RB is sequestering E2F. Yeah, green is E2F. So RB is sequestering E2F. Is growth factor signaling on? No. But look at the right. When growth factor signaling is on, then RB changes its configuration. It's phosphorylated. And E2F is what? Green is what? E2F is. When there's growth factor signaling, E2F will be released. But when there is no RB function, E2F will always be. So are you clear with that? RB sequesters, please keep that in mind. RB sequesters E2F when there is no growth factor signaling. RB releases E2F when there is a growth factor signaling. There is no RB, so the E2F will always be released. Virus, only do Epstein-Barr virus because Burkitt's lymphoma is because of Epstein-Barr virus also and because of chromosomal translocation also. No, understand that. Burkitt's lymphoma, I'll talk about that, but Epstein-Barr virus is related to Burkitt's lymphoma and cervical cancer because that is routinely done. Now, how does virus cause cancer? They cause chronic inflammation that damages mitochondria, that leads to cancer. They create reactive oxygen species because of mitochondrial damage that can lead to cancer. They also encode oncoproteins. Look at this. E6 is blocking P53. E7 is blocking, which one? E6 is blocking P53 function and E7 is blocking which function? RB. So they also damage or they counter the function of the tumor suppressor gene. So what does viral oncogenes do? Number one, they cause viral infection, cause chronic inflammation. That is not good for cancer. It causes mitochondrial dysfunction, reactive oxygen species, and encode oncoprotein. What is E6 doing? E6 oncoprotein is disrupting P53 function. Is it good? Or is it favorable for cancer? E6 blocking P53 function, is it favorable for cancer? Yes. And E7 blocking RB function, is it favorable for cancer? So that is how they can be cancers. Now, what do you need to know about this one? Please know APC is negative to beta-catenin. Very, very important. APC is a gene. It is a tumor suppressor gene. So it's loss of function or gain of function. So you should know all that in blue. Remember RB, P53, APC. So was APC tumor suppressor or proto-oncogene? Was APC tumor suppressor gene. So loss of APC gene leads to colon cancer, which is known as familial adenomatous polyposis colon cancer. But APC, what is the normal function of APC? It inhibits beta-catenin. What is beta-catenin doing? Beta-catenin promotes cell proliferation by MYC. Now, this MYC is not growth factor signaling MYC. Did you see MYC in growth factor signaling? Did you see MYC in growth? Was MYC at the end of growth factor signaling? Was MYC at the end of growth factor signaling? Yes, but this MYC of beta-catenin is independent of growth factor. It's different. So what I'm telling you, APC inhibits what? APC inhibits, you should know, beta-catenin, and beta-catenin promotes MYC and cell proliferation. Are you clear first with that? Now, before going to the answer, let me explain you the answer too, though I wanted to go to the slide. Now, Wind signaling is favorable for beta-catenin. Who is not favorable for beta-catenin? Who is negative to beta-catenin? APC. And who's favorable for beta-catenin? Wind signaling. Right? So Wnt signaling is normally favorable for beta-catenin and cell proliferation. In the absence of Wnt signaling, if beta-catenin is active, what is the reason? Again, I'm telling you, this is what you need to know. Wnt signaling, favorable to beta-catenin, cell proliferation. APC, inhibiting beta-catenin, not favoring cell proliferation. So in the absence of Wnt signaling, how can beta-catenin be active? Because Wnt signaling favors beta-catenin. Loss of APC. Did you understand that? So you should know APC inhibits what? Okay, first you should know beta-catenin is for cell proliferation through MYC. First, do you understand that beta-catenin activates cell proliferation through MYC? Yes? But APC inhibits what? Beta-catenin, and Wnt signaling favors? Wnt signaling favors beta-catenin. So how in the absence of Wnt signaling, beta-catenin can still be active if you lose APC gene, and that's exactly in the cancer. But I'll explain you with the diagram. That would be more easy. This is looking a bit complicated, but are you clear with that? APC inhibits beta-catenin, and Wnt signaling favors beta-catenin, and beta-catenin is for cell proliferation through MYC. Are you clear with that? Now let's look at this. At the bottom of the cells, you need proliferation. At the top of the cell, you need differentiation, no proliferation. So at the bottom of the cells, do you want Wnt signaling to be on? Bottom, you need proliferation. Top, you need differentiation. Yes. So that's why at the bottom, Wnt signaling is on, and Wnt signaling inhibits what? APC. And what does it activate? Beta-catenin. But at the top of the crypt, do you want Wnt signaling to be on? At the top of the crypt, do you want Wnt signaling to be on? No. So APC will be active at the top of the crypt. Will APC be active at the top of the crypt? So at the top, there is no cell proliferation, but cell differentiation. That's what you want. You want now to differentiate this cell for absorption in the colon, for its function, in the colon, right? So do you understand that? Let me explain you again. But are you clear? Or you can say that Wnt is negative to APC, yes? And APC is negative to Wnt is negative to APC, and APC is negative to beta-catenin. Are you clear with this line diagram? Don't flip-flop that. Now let's understand that. Familial adenomatous polyposis, do you lose APC? Yes. And HNPCC Lynch syndrome, is it same as FAP? Is it same as FAP? Which genes are lost here? Not APC, but mismatch repair. So are you aware of both the colon cancer, different genes? Don't flip-flop them. Where is endometrial cancer in female? FAP or HNPCC Lynch syndrome? Endometrial cancer in female, HNPCC. When you see endometrial and colorectal together in female, think about Lynch syndrome, not BRCA1, BRCA2, right? Are you clear with that? Beta-catenin stimulates proliferation, please. You should know that. They also use MYC, but this is not the MYC of growth factor signaling. Are you clear with that? Wnt signaling inhibits APC, and APC inhibits beta-catenin, and beta-catenin stimulates cell proliferation through MYC. This is not the same MYC of growth factor signaling. So at the bottom of the crypt, do you want Wnt signaling? Do you want? Yes, because you want beta-catenin to be active and cell proliferation. But at the top of the crypt, do you want Wnt signaling? No. So what is on at the top? APC and no cell proliferation. So tell me, loss of function of beta-catenin or gain of function of beta-catenin will lead to cancer, colon cancer. Loss of function of beta-catenin or gain of function of beta-catenin will lead to colon cancer. Loss of function or gain of function of beta-catenin, not APC, I'm asking, beta-catenin. I've told you the function. Gain of function of beta-catenin and loss of function of APC will lead to colon cancer. But Don't flip-flop that. In familial adenomatous polyposis, there is loss of function of APC. That's the initiating event, not gain of function of beta-catenin. I just asked you that question, right? But what is the cause of colon cancer? Loss of function of APC at the top, where you don't need proliferation. You actually should have differentiation, right? Does that make sense? Any confusion? I will come back and redo it. I just want to hit the key points. So 2 more minutes. Now, chromosomal translocation. This is very, very important, right? So are you clear with the bottom of the crypt proliferation? Beta-catenin should be active or inactive? Bottom of the crypt. Wind signaling should be active or inactive? Bottom of the crypts. Wind signaling. Active. As you go on the top, wind signaling should be, APC should be active or inactive as you go on the top. APC should be active or inactive? Top. No. Again. Oh, no, I'm saying active. Okay. Please, this one, last thing, okay? Wind signaling should be active in the bottom so that beta-catenin is active and proliferation. Yes? Yes? I ask you at the top of the crypt, should wind signaling be on? Top of the crypt. So who is on here? Is APC active? Is APC active at the top? Yes. When you lose APC, then you get cancer. So when I asked you, normally I'm asking you. So please know that wind signaling is negative to APC. APC is negative to beta-catenin. So that's why I ask you at the bottom of the crypt, wind signaling should be active for beta-catenin and cell proliferation. At the top of the crypt, wind signaling should not be active. What should be active here? What should be active here? Wind signaling or APC? APC to stop proliferation and start what? APC should be active on the top to stop proliferation and start what? Differentiation. When APC is lost, look at the top. What do you get? Tumor, polyps, cancer. So the top APC is active and the bottom wind signaling is active. Wind negative to APC, APC negative to beta-catenin. Please be mindful of that. This is very, very important. Please go back and do it. Okay. I explained you. Now, chromosomal translocation, last thing I will do with this only. This is my last slide. Chromosomal translocation results in 2 things. Either wild gene is under strong promoter. Remember this? Wild gene under strong promoter. And this is fusion gene. Don't do Ewing sarcoma. Only CML and promyelocytic leukemia. Let's rule out all the answer. This will be my last slide. Let's do that. So they are saying acute promyelocytic leukemia. Let's rule out all the answer. First of all, overexpression of transcription factor. Do you know which they are talking about? Which transcription factor I told you to know very well? Which transcription factor I told you to know very well? Let's finish. MYC. Now, where is MYC overexpressed? Burkitt's lymphoma. Burkitt's lymphoma is 814 translocation, where 8 MYC gene comes under the strong promoter. Now here MYC gene is amplified or overexpressed? Burkitt's lymphoma. Because MYC gene is coming under strong promoter. Burkitt's lymphoma. 8 is MYC. In chromosomal translocation, you should know the translocation chromosome, gene involved, and what function altered. So Burkitt's is 814. 8 gene is MYC. 14 is heavy chain promoter. MYC come under the influence of heavy chain promoter. So in Burkitt lymphoma, MYC will be overexpressed. So A should be the answer for which chromosomal translocation? Burkitt's or follicular? Burkitt's, follicular, CML, or promyelocytic leukemia. You should know only 4. I'll rule out all. A should be the answer. Burkitt's, follicular, Burkitt's. Please know that. Note it down. Note it down. A should be the answer of Burkitt's. Let's look at B. Overexpression of anti-apoptotic gene. What's anti-apoptotic gene? FAS or BCL2? BCL2. Where is BCL2 overexpressed? Follicular lymphoma. It's 14-18 translocation. where BCL2 gene is now under strong promoter and BCL2 is overexpressed. So where is MYC overexpressed? Where is MYC overexpressed? Which is answer for 1, Burkitt's. Where is BCL2 overexpressed? Anti-apoptotic. So B should be the answer for. B should be the answer for. B should be follicular lymphoma. Let's go C. Where is the I think production of constitutively active could happen anywhere. C, I will rule out. Let's go to E. Intracellular tyrosine kinase active is CML. What is CML? 922 translocation, which forms a fusion gene known as Philadelphia chromosome, Abelson BCR fusion gene. Abelson is 9, BCR is 22. What's the outcome of that? Here, gene is not overexpressed. The fusion gene is oncogenic. What is the oncogenic here? It's intracellular, the E. E is the answer for, it's not receptor tyrosine kinase. The intracellular tyrosine kinase is turned on in which one? Here there's no overexpression. The intracellular tyrosine kinase is turned on and on and on. So which is this one? CML. So E should be the answer for CML. A answer should be Burkitt's. B should be follicular. Now let's look at this nuclear receptor. That is AML or PML. PML or AMLM3 is the same. This is 15-17 translocation. 15 is PML gene. 17 is this retinoic acid receptor nuclear gene. So this is the answer for acute promyelocytic leukemia, which is 15-17 translocation. 15 is PML, 17 is RAR. Remember, retinoic acid is a nuclear receptor. So that is fusion gene is oncogenic. PML, RAR is oncogenic. So D is the answer for PML. So that is why your answer should be D. And that's all for, oh, sorry. That's all for today. Okay. I wish you good luck. Great.
I have a good question, Dr. Bob.
Sure.
For C, is it any of these, like sarcoma?
C is like, no, I would say C is like, could be a mechanism of any cancer. That
Okay,
there,
just
remember,
general.
there's a receptor tyrosine kinase and intracellular, but I think the translocation word should come to
Okay.
call it CML. That's why I didn't pick C.
Okay, perfect.
Okay.
Thank you.
Okay, great. I wish you good luck. Yes, you enjoy your little break too. I'm glad it helped. I redid the session, but I'm glad. Great. I wish you all good luck. If you have any questions, otherwise, we are good with the session. Take care. Bye-bye. Everybody good here? Great. Take care.
Dr. Bob,
Yes, sure.
how many questions are going to